All right, team. Here we are again, still talking about bacteria, using that term loosely to apply to all prokaryotic organisms. Reproduction is the next topic, and primarily these organisms are going to reproduce asexually. Now, remember, um, these organisms don't have a nucleus. And so you start thinking about things like mitosis. Well, mitosis can't really happen um, because there's no nucleus. So the most common way that they would reproduce, if you have bacteria, and remember it has uh, typically what we like to think of as circular DNA. It's a little circle of DNA. And what it will do is it will replicate its genetic material. My colors are all mixed up. I apologize. The colors are kind of irrelevant at this point. It will replicate its genetic material, make two copies of it, and then ultimately this thing called simple fission occurs, and you can see that right here, simple fission, where you basically are just going to then divide the cell into two pieces like this. Each one gets the genetic material, and then ultimately have two little baby bacteria. Okay, and so here's the thing though, the replication process in bacteria is not nearly as sophisticated as it is in eukaryotic cells, like in us, for example. And because of that, mutation occurs a lot. And, you know, we believe mutation in eukaryotes, in us, for example, occurs in about rate of about 1 in a million replications. In bacteria, it's more like 1 in 100 to 1 in 1,000. So every 100th cell might have a little glitch, and we could draw that, you know, by using a different color right here and showing, well, they're supposed to be identical, but actually, you know, this one could have a little slightly different genome sequence now. So the single greatest source of genetic variability in the prokaryotes is mutation during the replication process of the DNA, or at any point. Okay. No sexual reproduction as we know it. Think about what sexual reproduction means. Meiosis and fusion. Well, these organisms don't have a nucleus and they don't really have chromosomes, so they can't really do meiosis. And so we don't think of bacteria as reproducing sexually. Okay, the closest thing that they come to in terms of sexual reproduction, it's not technically sexual reproduction, but it's this thing called conjugation. And this is a very interesting phenomenon. You can have two bacteria and they'll actually form what's called a conjugation tube. Remember those little things called pili are those little finger-like extensions. Well, they can form this conjugation tube between them like this. And let's just say they're the same species, but we'll draw, you know, they're individuals of the same species, let's say, but we'll draw their genomes different colors. What they can literally do is they can pass little bits and pieces of their genetic material between the two individuals, okay? And they're kind of, this is a way of sort of shuffling up the genes. It's not sexual reproduction per se, but it's a way of shuffling the genes. Typically, bacteria are more than happy just to reproduce asexually until the environment starts to get bad, and that could mean anything depending on the species. But when the environment starts to go south, this is when they'll often form these conjugation tubes and exchange genomes. Now, one of the things that we have learned, which is very bizarre, is that these tubes can actually form between different species of bacteria. So maybe this is species A and species B. And this is pretty bizarre because they're not the same species, but they're exchanging genes. And one of the things that we know is that some of the antibiotic resistant strains of staph, for example, obtained their antibiotic resistance from harmless bacteria that live in the gut that were actually resistant to staph and so, or to antibiotics rather. And so they passed these resistance genes on to the pathogenic bacteria. And this is pretty scary stuff if you think about it. Okay. All right. Moving along. Now we get to the cyanobacteria. And this is a special class of bacteria. And what's special about them is that they are, in fact, autotrophic. And we had this discussion, earlier about the ways that organisms make their livings. We are heterotrophic organisms. So autotrophic organisms are typically photosynthetic, although they could be chemosynthetic. So yes, there are, in fact, photosynthetic bacteria. Okay. And what we see is that the terms that we were using when we were talking about heterotrophic bacteria don't particularly apply to the autotrophic ones. For example, words like bacilli and spirilla and things like that. We don't use those to describe these. One of the things that we see, there's two sort of what you might consider advancements that we see in these organisms. They can be unicellular, but more typically, they are either colonial, and these are life forms. So all of the heterotrophic bacteria, all the ones we've talked about thus far, are unicellular. When we get to the autotrophic group, the photosynthetic ones, they can have slight advancements. For example, they can be colonial. And you can see here, we have a colony of these bacteria. And so what's a colony? Well, it's some individuals here living, in this case, in a common matrix. It's like a gelatinous matrix. So that would be an advancement in life form. And then even more common is filamentous. And there's a really good example of long filaments or chains of cells. And we can see that here as well and here as well. Okay, so, what does filamentous mean? Well, what it means really is that cell division, in this case, simple fission, can only occur in one plane. And so here's a filament with a bunch of cells. And when cell division occurs, it can only occur in one plane like this. What this means is the organism can never get any taller, or any thicker. It can only get longer. And so that's what filamentous means. And so we're going to see some examples of that. Now, the other thing to consider is we also have cell specialization. We haven't seen this yet. All of the heterotrophic bacteria, one cell is just like the next. There's no specialized cells. Well, now that we have a filamentous life form, we can have certain cells in there that are specialized, and that's what we're going to see. Now, these organisms are ubiquitous. They're everywhere. When you see green, slimy, green-looking stuff growing in a mud puddle, let's say, or in an abandoned swimming pool or a pond, oftentimes you think it's algae, but it's actually not algae. It's cyanobacteria. And this group of organisms has in the past been called the blue-green algae. Oh, I cringe when I hear that because they are not algae. Algae are heterotrophic. Are eukaryotic organisms, and these are very much prokaryotic. So don't call them algae. Good example of this being misused is there's one called spirulina. And some of you have maybe taken spirulina as a supplement. You might have eaten it in a green smoothie. And spirulina is often called blue-green algae. It's a bacteria, it's not an algae. So just keep that in mind. So we won't worry about this word here. Oops, shoot. Now look what I've done. We won't worry about this word aconite. I'm not going to hold you to that word. So you don't have to know this word. An aconite is a resistant spore. Now, we see a more common word is endospore. And we see endospores in both the heterotrophic and autotrophic bacteria. And what an endospore is literally a resistant stage in the life cycle. We don't know how long these spores can be viable. We know at least millions of years and maybe longer. The one specialized cell that I want you to know, though, is this thing called a heterocyst. And the heterocyst is a specialized cell where a particular chemical reaction occurs, and that particular chemical reaction is called nitrogen fixation. I know it sounds like some sort of neurosis that a human might have, but it's not. It's actually a very important and useful chemical reaction. And the chemical reaction involves making nitrate from nitrogen gas. And so we can take a look at that, and we'll look at some specializations here. Let me get rid of the drawings. Okay, so you'll notice here's the chain of cells in the filament, and notice this one cell right here looks different. This is a heterocyst. Okay, it's that specialized cell where nitrogen fixation is going to occur. Now, this brings us into the topic of talking about this idea of a symbiosis. Okay, and in this case, the symbiosis is between two. A symbiosis is always a relationship between two organisms. In this case, it's a relationship between vascular plants, typically legumes, and I'm just going to put legumes, and cyanobacteria. Now, what are legumes? Well, things in the bean family: beans, peas, clover is a legume. Okay. Here's the thing about this word symbiosis. It's very misleading in a sense, because when people see this word or hear it, a lot of people automatically assume that it's somehow beneficial for both of the organisms. And that's not necessarily the case. And so I want to be clear about that. For example, a parasitism is an example of a symbiosis. And in a parasitism, well, what do you know? One benefits at the expense of the other, right? The point is that this word symbiosis does not tell you anything about the nature of the relationship. It just means there's two different species of organisms that are living together. By living together, we mean they're physically touching one another, okay? So there's parasitism is an example of a symbiosis. There's one called commensalism. And in commensalism, one benefits and one is unharmed. So you might see, for example, the Spanish moss hanging in a tree. The Spanish moss is clearly benefiting from the relationship. The tree is basically unharmed in the relationship, okay? And then there's mutualism. And in mutualism, both benefit. So in this particular symbiosis, it turns out it's a mutualistic symbiosis, meaning both of these organisms, both the legume, the vascular plant, and the cyanobacteria are going to somehow benefit from the relationship. So anytime we talk about a symbiosis, we want to know what's the nature of the relationship, and who are the major players, and what are they each getting or giving, okay? So in order to understand this, we really have to understand a little bit about the chemical reaction. And you can see here, here's the chemical reaction. This is nitrogen gas. Oops. So this is nitrogen gas, N2. This is water, of course. And this is nitrate. Okay. It turns out, frustrating as it must be, that plants cannot use nitrogen gas as a source of nitrogen. They don't have a way of cleaving this molecule here into two pieces and snatching the nitrogen that they need to make things that are useful out of nitrogen. For example, the chlorophyll molecule has nitrogen in it. Okay. So here's the nature of the relationship. Let's draw a little clover plant here. I should be drawing it green, but I'm going to be lazy. Here's clover with its roots. Four-leaf clover, y'all. Okay. What you will see if you dig up a clover and look at the roots almost always is these little things called root nodules, and I'm going to draw those in green. And they look like these little balls like this on the roots of the clover. These are called root nodules. The nodule is actually made by the plant. If you cut it open inside of the root nodule, guess what's in there? A cyanobacteria. Okay. And so here's what happens. Inside these root nodules, the cyanobacteria is able to do this chemical reaction and turn nitrogen gas into nitrate. Nitrate is a source of nitrogen that a plant can use. And so now the nature of the relationship starts to become a little more clear, right? The cyanobacteria gives the plant nitrate. A usable form of nitrogen. Okay. What if it's mutualism though? Well, now we see what the plant gets, but what does the cyanobacteria get from the plant? It's got to give something. The plant gives two things: place to live on land, right? Because normally these cyanobacteria live in water. They would dry out very quickly on land. All of a sudden, now the cyanobacteria has a way to colonize land. That's a big deal. That's a good thing. In addition to that, it gives the cyanobacteria, which normally would be photosynthetic and could make its own sugar, but now it's in the ground, it gives it sugar. And so, you know, at first glance, this almost looks like a parasitism. You're like, well, this thing is just stealing sugar from the plant, right? But on closer look, you realize that it's giving the plant something back. It's giving the plant nitrate. And what we actually know about this relationship is it's really the plant that controls the relationship, even though it's a mutualism. For example, if you grow legumes in a very nitrogen-rich soil where they're not hurting for nitrogen, they have plenty already, they will not allow typically the cyanobacteria to form these root nodules on the roots. They're like, nah, we don't need you. So, but otherwise, you know, it's a very useful chemical reaction. Now, you know, one of the things you might consider doing if you're growing a garden and you're planting beans or peas for the first time in the soil, they sell inoculant, and the inoculant is literally this cyanobacteria, and there are different species, and so you would want to, you know, make sure you got the right one for your beans or your peas. There's different species for beans and peas, but the point is you sprinkle this powder that has this cyanobacteria on the roots or on the seeds rather, and then when you plant them, the nodules form almost immediately, and this benefits the plant greatly. Now, if you don't inoculate them, more than likely, eventually these cyanobacteria will find the roots and it'll happen anyway. But this is a way just to speed up the process. Okay, so just to reiterate, what are root nodules? Root nodules are these little balls that form on the roots of legumes, inside of which is a cyanobacteria, which is able to do nitrogen fixation, okay? Nitrogen fixation occurs in the root nodule. What is nitrogen fixation? Well, it's where these cyanobacteria take nitrogen gas in the presence of water, and they make nitrate, and they also release oxygen in the process as well. Okay, so that's one of many symbiotic relationships we'll talk about involving bacteria. We actually mentioned another one with the heterotrophic bacteria that was a mutualism, and the other one was the intestinal flora, right? The bacteria that live in your gut. Okay. All right. Oh, so now we get to talking about viruses. And here's the important thing about viruses. The first thing I want to mention so that I don't forget is that they are not living. They do not fit the criteria of life because they have no metabolism. And what that means is anytime energy is involved with respect to a virus, the virus has no way of making its own energy. It can only use the energy of its host. And so because of that, because it doesn't fit into this sort of artificial, granted, but nonetheless, we have these criteria of life, and it doesn't quite fit, and so we don't consider it alive. Instead, we consider it, oh, I'm not writing very well. Look at this mess. We consider it typically, oh gosh, we consider it to be an infectious particle. There might be an I in there. I think there is. Okay. So think of viruses as an infectious particle. What do they consist of? Well, notice it says nucleic acid. It doesn't say DNA or RNA because it can be either one. There are DNA viruses and there are RNA viruses. For example, COVID-19 is an RNA virus, as is HIV, the virus which causes AIDS. So there's a little fragment of genetic material, and we're just going to call it nucleic acid. It's one or the other. I should put an or here, not an and. It's one or the other, DNA or RNA. And then the other thing that it has is a protein coat. Okay, so, these little tails are often how it can attach to its host cell. But for our purposes, think of it like a little fragment of genetic material surrounded by a bunch of proteins. Okay. So what we want to understand about viruses is, how do they infect? How does our immune system deal with them if you're talking about a pathogenic virus on humans? We want to understand that there are viruses which infect all living things. All eukaryotes are subject to some viral infection. Plants get viruses, animals get viruses, etc. So nothing is immune to viral infiltration. Okay, so we classify it as an infectious particle. They're often named after, we don't use the Linnaean system to name them because they're not living. They're not put in a kingdom, for example. They're often named after places where they've been found or by what they do as pathogens, for example, Ebola virus or human immunodeficiency virus. Those are two good examples. Or you'll often see numbers like, oops. Sorry. You'll often see numbers like, letters and numbers like H1N1, for example. So if you see something like H1N1, this is telling you something about the genetic sequences, and this would mean something to a geneticist or someone that studies these things. Okay, so there are lots and lots of different kinds of viruses which infect humans. They infect different parts of your body. And so we, this is a short list. Obviously, there are lots of other ones, but we can start here, and we're going to touch upon a bunch of the really important viruses which infect humans. And anytime you have something like a virus or a bacteria that infects humans, we say that they are pathogenic, right? If they're pathogenic, that just means they can infect you and make you sick. So we start with cutaneous. Well, what is this? This is skin, right? And so measles, rubella, chickenpox, and smallpox, these are all cutaneous viruses. Measles, we fortunately have vaccines for these. For example, smallpox killed and crippled many people until a vaccine was developed, right? So, when was the last time you worried about smallpox? Well, probably never, because of the vaccine. Same with polio, okay? So these are so-called childhood diseases as well, and measles, mumps, rubella, chickenpox. These are all things that many people would be vaccinated against. Okay. It turns out that if you get these later in life, it can be more serious. For example, measles can kill you if you get it as an adult. It can be very dangerous. Gastrointestinal, these are the so-called stomach flus. Oh, the dreaded stomach flu, right? Noro and rotaviruses. These are things which wreak havoc on cruise ships and daycare centers and stuff like that. They're typically short-lived, usually about 48 hours, but during that time, the individual is very ill, vomiting and fevers and chills and vomiting. It's a miserable thing. And, luckily it doesn't last very long. If it lasted much longer, it would kill you, right? Because you would lose so much fluid and become dehydrated and loss of electrolytes. Okay. So these can be foodborne, they can just be typically on the surface. The thing about these viruses is you get them by ingesting the virus, and then it passes its way. The viruses are very resistant to the stomach acid, and then they pass into your intestines where they do their thing. Okay. So respiratory viruses, influenza and the common cold are the two most common viruses on Earth, you might say, in terms of respiratory viruses at least. So, we have a flu season, for example. We'll talk a little bit about immunity and how your body deals with things like the flu and the common cold. The common cold is a type of coronavirus, and of course, that is also true of SARS and COVID-19. SARS stands for sudden acute respiratory syndrome. We know now that COVID-19 affects multiple organs, but because when it becomes lethal, it's typically due to its infection in the lungs. We still consider it to have a respiratory component. And at the time of the making of this video, we are in the middle of a global pandemic of COVID-19. It's unprecedented in modern times. So, there's that. Now, just so you know, there have been influenza pandemics in the past. In 1918, there was the Great Flu Pandemic, which actually started in the United States. It started in Kansas, they believe, and spread throughout the world. It's estimated that 60 million people died from this one influenza strain. The scary part about these strains, when you have a new strain of influenza, for example, no human has ever been exposed to it. It's a brand new virus. No one has resistance, theoretically. So this is why it killed many, many people, 60 million people. At the making of this video, just in the US, we just passed 200,000 deaths from COVID-19 in the USA. That's a lot of people. That's everyone in the county of Buncombe where I live twice, that's a lot of people. Okay. So then we get to the hepatic viruses, hepatitis A, B, C, D, and E. Hep C being a particularly difficult one to deal with, and it becomes chronic and difficult to get rid of. They do now have treatment for it where they can rid your body of it. Um, because the liver is such an important organ, anytime the liver is involved, you're going to have issues, right? And so Hep C is one that can become debilitating and ultimately kill you. Now, it is spread typically through blood contact. It's not considered a sexually transmitted disease per se, but exchange of blood. So obviously, if you're a IV drug user, you're going to be at high risk. There was a large population of relatively affluent individuals that developed Hep C in the 80s and 90s, and they're like, what is going on with these people? And what they discovered was that they most likely got Hep C from snorting cocaine, if you can believe that. The straw that they were using and passing among around in a circle as each person snorted their line of cocaine, the cocaine was bursting the blood vessels, the blood capillaries in their nose. So there would be tiny trace amounts of blood on the straw as you pass it from one person to another. And they believe this is how Hep C was being spread back then. So you got to watch out about things like that. Okay. Then we get to STDs. Wah, 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 right? HPV is, we often use abbreviations for these, human papillomavirus. Human papillomavirus is the virus which causes, there are, I think, 180 different strains of human papillomavirus. It can cause, for example, plantar warts, which are just skin warts. It can also cause genital warts, right? Now, the thing about HPV is that most people that get HPV are asymptomatic. They don't have any symptoms. And so if you get human papillomavirus, if you get the genital virus, you probably don't have any symptoms and you would never even know you have it. It's incredibly common. It's estimated that 90% of humans have HPV, right? As soon as you become sexually active, you more than likely would become exposed to some of these strains of HPV. And the scary thing about them, though, is that there are some strains, particularly 16 and 18, which are linked to cervical cancer, right? In fact, they're not only linked to cervical cancer, but no human that we know of, no female has ever gotten cervical cancer before first being exposed to one of these strains of HPV. And so now we have a direct link between a virus and cancer. And this is well documented, okay? So what that means is if you're exposed to these viruses, you greatly increase your risk of cervical cancer. It increases your risk. Now, men are not immune either. It also increases the risk of penile cancer, anal cancer, and throat cancer. And the throat cancer is a really big one, y'all. There's a great uptick in throat cancer, particularly among men, and this is believed it's due to oral sex, right? And so you can give oral sex to a woman that has HPV 16 or 18, and then you can get an infection in your throat and then later develop cancer, sometimes 8 or 10 years later. So it can be a long period of time before the cancer develops, but nonetheless, this is something that you have to think about. How can you protect yourself? Well, there are vaccines now for these particular strains. And they used to just say the vaccines were for children, but they're not saying that anymore. They up to, I think, the age of 40, you can get these vaccines. And so this might be something that you might think about if you have been, if you just want to get the vaccine, unfortunately for men, there's no way to test for these viruses. There's no way to test your throat for throat cancer. But as many of you women know, if you have, when you go to the gynecologist, they'll do a pap smear and they screen for cervical cancer in the cells of your, of your pap smear. And if they detect these HPV strains, the genetic material of the HPV strains, they will let you know, you have this particular strain. Most individuals clear it within 2 years, and that doesn't cause them any trouble. So I don't want you to think that just because you are exposed to HPV, you're automatically going to get cervical cancer. It doesn't work that way. And again, fortunately for women, there's a screening. And so cervical cancer is actually becoming more and more rare because we're screening for these viruses and then doing treatment to prevent them. Okay. All right, and then there's herpes, which is HSV, herpes simplex virus 1, which is oral, and 2, which is genital. Okay, well, first thing I want to tell you about herpes is that although HSV1 is called oral herpes and prefers the mouth, it can go down there. And although genital herpes prefers the genitalia, it can go up there. Okay, so what that means is if you have an oral herpes lesion on your mouth, don't be going down there on your sexual partner. And if you're having a genital herpes outbreak, don't go up there. Don't go down there on your sexual partner. Okay, so they can be transmitted from the genitals to the mouth or vice versa, although they do have their preferences. Okay, how common are they? HSV1, incredibly common. About 80% would test positive for oral herpes, and the vast majority of them would be asymptomatic. Genital herpes, are you ready for this? Oh my goodness. 1 in 4 females and 1 in 6 males would test positive for genital herpes. Now there is a blood test. And so there's a blood screening test they can do now, and that's where these numbers come from. It means that you have, the antibodies for these 2 different strains, 1 or 2. It used to be they couldn't tell the difference, and now they can. Well, you would think that they would be testing people like mad, but they're actually not. And a lot of it has to do with the fact that 1 in 4 females, about 25% of all females would test positive for HSV2 for genital herpes. But of those positives, only about 20% of them have any symptoms. 80% of them are asymptomatic, meaning they don't even know they have it unless they were tested. Okay. And the problem is there's a stigma associated with it that quite honestly can be very dramatic to the psyche of these individuals when they find out, even if they're asymptomatic. It creates this, literally there have been cases where people have committed suicide because they had herpes and they were so upset about it, or at least that was, what they said. So, what can we do? Well, it's challenging because of the asymptomatic aspect to avoid. You can be an asymptomatic carrier, meaning you can have no symptoms and you can still transmit it to someone who can become infected, and they may have symptoms. There's no way to know. They may develop symptoms from your asymptomatic virus. Okay. So, one of the things I would say is you can, if you're concerned, you can get screened. You can certainly have your sexual partner screened. If you do have an active infection and you're symptomatic, obviously you would want to avoid contact during that period. And then there are also antiviral drugs that can be used to treat it. And then there's HIV, which we're going to talk extensively about, human immunodeficiency virus, in just a bit. Neurologic polio and rabies and viral meningitis, anytime, your brain is involved, very serious illness, brain and spinal cord, polio affecting the spinal cord, and it was a crippling disease prior to the advent of vaccine. And again, when was the last time you worried about getting polio? Well, you didn't because of the vaccine. Keep that in mind, okay? Rabies, there is a treatment for rabies. It's a horrible illness if you should contract it. Typically, you would contract it from being bitten or coming in contact with a rabid animal. It could be any kind of mammal. It could be a bat. It could be, a raccoon, a dog, whatever. Okay. One of the ways they test animals for rabies, if you're bitten by them, is to examine their brain. And so if you're bitten by an animal and you euthanize the animal later or kill it if it's a wild animal, try to preserve its brain so they can test it. Otherwise, you may have to have the treatment. Viral meningitis can be spread by mosquitoes, and it can be very serious illness. The meninges are the coverings of the brain. When they swell up, it puts pressure on your brain and it can cause brain damage. And then lastly, we have the hemorrhagic fevers, as they're called, Ebola, dengue, yellow fever. These can be very serious. Hemorrhagic meaning you're going to bleed, your capillaries are going to bleed. When your blood capillaries bleed, it's very serious, right? And so Ebola is one of the so-called hot viruses, which is 70 to 80% fatal if you get it, but it's obviously not very common. Dengue fever is spread by mosquitoes, and we have outbreaks in tropical areas now and again. It can be very serious. It can kill you. There are different strains. A recent study just came out that showed that perhaps being exposed to dengue fever and having recovered from dengue fever might protect you some against COVID-19 because your immune system is recognizing these different viruses. It's recognizing some of the proteins on the COVID-19 virus based upon the dengue fever, even though they're very different. And then yellow fever is another hemorrhagic virus. Okay. So what we're going to do is talk a little bit about HIV. But before we do that, I want to talk a little bit about how viruses infect. I think it's important to understand how they infect, and then what you can do, or not necessarily what you can do, but what your immune system does to then counteract this infection. So it turns out that, actually, let me just get a whiteboard. This will be easier on a whiteboard. It all has to do with antigens. And by antigens, you could kind of think of the antigens, if this is a bacteria or a virus, it could be a bacteria or a virus actually, but we're going to let this be a virus. And there's its little piece of genetic material. And this virus, you think of the protein coat that's on this virus as kind of like a flag. Okay, it's flying a particular flag, and this flag represents the antigen complex. Remember, there are proteins of this particular virus. Now, keep in mind, a different virus is going to be flying a different flag. They're not going to have all the same proteins, right? So along comes then a white blood cell, like a killer T cell. It's flying the flag of the self, right? And we'll let this be a killer T lymphocyte. And what these killer T lymphocytes do, they literally roam around the body looking for things that don't belong. How do they know it doesn't belong? They look at its flag. Before you're even born, your immune system is developing, and these cells, your immune system starts to make a list of self proteins. Right? So this represents the self antigens, all the antigens that your body makes. When it looks at something that's in your body and it starts looking at these proteins and comparing them to the self proteins, when enough of them don't line up, it's like, whoa, wait a minute, this thing doesn't belong. Now, one example of this that's sort of can have a negative effect is when these lymphocytes react to things that are harmless, like a pollen grain, for example. So many of you may have allergies to dander or pollen. And in that case, the lymphocyte is reacting to the antigens of something that's harmless, right? And your body can overreact to that. But in this case, it's reacting to a virus, if we're saying this is a virus. So what then happens is the killer T cell's job, literally, is to come and engulf this, right? Endocytosis, right? And so then this virus ends up being engulfed, and it would be then inside of this killer T cell, where it would then be destroyed, right? It would be got rid of. And so you could see this is a cell by cell process, which would be relatively slow, T-O-S-I-S, sorry. Be a relatively slow process, one killer T cell attacking one virus. But something else happens. When this killer T cell starts recognizing this foreign antigen, it causes a whole nother part of your immune system to react. And this involves activating other cells which produce antibodies. And also produce memory cells. Antibodies it's almost like spray paint. It labels the virus. The antibody fits the antigen of the virus and basically neutralizes it. You could think of it like that. So what do antibodies do? They neutralize the antigen. Okay. What do memory cells do? They remember the antigen complex. What that means is for every virus you've ever been exposed to, there are memory cells floating around in your blood. They remember the antigen complex. And so if this same virus ever gets in your body, or one that's similar enough to it, the memory cells will immediately destroy it before it has a chance to even make you sick. Okay? This is the whole principle of how a vaccine works. When you're injected with a vaccine, you trick your immune system into the antibody and memory cell response by using either an attenuated, which means a weakened version of the virus that can't actually make you sick, or by just stripping those antigens, those proteins, away from the genetic material and injecting those in. So right now, at the making of this video, we're working on a vaccine for COVID-19. And what they've shown so far in phase 2 is that we are able to generate very strong antibody response in humans. And so we're all very hopeful that this is going to work out, right? And that we're going to have a successful vaccine so that we can get on with our lives, so to speak. Okay. So, this is basically how the process works. A virus infects cells. That's the other thing that we'll talk about here in just a second. How does the virus infect? Well, let's look at that. We'll let this be a host cell with its nucleus. and we'll let this be a virus. And the first thing that occurs is that the virus attaches to the host, like so. There's its little genetic material. And typically the way that it attaches is that the host cell has to have some sort of complementary shaped protein to the viral protein on these little tails of the virus. And so there's this little attachment point. If the host cell lacks that attachment point, the virus can't attach, it can't infect. But if the host does have these attachment points, the virus is able to attach. At that point, the virus injects its little piece of genetic material into the host. And at that point, this part is kind of irrelevant. It's just a bunch of proteins. It's just an empty shell in a sense. That little piece of genetic material then makes its way through a nuclear pore and into the nucleus of the cell and becomes incorporated into one of the chromosomes of the host cell. At that point, that little piece of genetic material takes over. And what it does literally is it forces the host to make many, many copies of the viral genome. And so pretty soon, this host cell is full of viral genetic material. Okay. If that's not enough of an insult, at that point, it signals to the ribosomes to start assembling the protein coats around, make the proteins to assemble around each little piece of nucleic acid like this. At this point, how's this host cell doing? Well, it's not doing so great, right? It's now swollen with viral particles. Lysis occurs, it splits open, and out they come. And where are they going? To infect another host, right? And so we talked about, initially this can overrun your system, but then the killer T cells come into play, and they start finding these things and recognizing them as being foreign. And then the immune system kicks in, antibodies are produced, eventually your body clears the virus or not, depending upon the virus, okay? So this gives you an idea of how viruses infect. And I think we'll stop there because we want to, we'll finish up viruses in the next video. We'll talk in detail about HIV and how it infects and all of that kind of thing next time. And I hope you have a great day. I'll see you later. Bye-bye.